Welcome, class. Settle in because I am absolutely thrilled about today's session. In this special masterclass edition of our Explainer, we are diving deep into the awe-inspiring and sometimes terrifying mechanics of cellular division. We're going to explore that incredibly razor-thin line between meticulous biological order, the literal mechanism that builds and maintains our bodies, and the total, devastating rebellion of cancer. Let's get right into it.
To set our academic foundation today, we actually need to rewind all the way back to 1855. There was this brilliant German physician, Rudolf Virchow, and he laid down an absolute biological rule. In Latin, he called it omnia cellula a cellula, literally, every cell from a cell. The entire continuity of life is completely based on this one idea. A cell doesn't just magically poof into existence out of thin air. It's the direct descendant of a pre-existing cell, passed down in this unbelievable, unbroken chain of replication that stretches all the way back to the very dawn of life on Earth.
So, here is our syllabus for today's masterclass. First, every cell from a cell. Then, phases of the cell cycle. Third, the dance of mitosis. Fourth, the checkpoint control system. And finally, when cells go rogue.
All right, kicking things off with Module 1, every cell from a cell, the foundational law of continuous life.
Just think about the immense physical task a cell has to pull off when it passes on its genetic material. Before it can divide into two identical daughter cells, it has to completely copy its entire genome. And honestly, the sheer scale of what we are talking about here is just staggering. A single, completely microscopic human cell contains about 2 meters of DNA. That's a length about 250,000 times greater than the cell's actual diameter. Imagine trying to pack 2 meters of incredibly delicate, absolutely vital string into a microscopic, invisible sphere without tangling or breaking it. I mean, it is a breathtaking feat of biological engineering, isn't it?
So how does it manage this without making a mess? Well, to deal with that staggering length, the cell uses a brilliantly ingenious packaging system. It takes that loose DNA and tightly coils and folds it around specific proteins. This whole complex, the DNA and the proteins working together, is what we call chromatin. Now, when it's go time, when it is actually time to divide, this chromatin condenses even further. It becomes densely coiled and superfolded into those distinct, thick structures we all know as chromosomes. This intense packaging ensures that when the cell finally splits, that massive 2 meters of critical information doesn't get ripped, sheared, or just hopelessly tangled up.
Moving right along to Module 2, Phases of the Cell Cycle, which is all about the timeline of growth and division.
There is a really common misconception out there that our cells are just constantly, endlessly splitting in half all day long. But the reality of the timeline is completely different. A massive 90% of the cell cycle is entirely dedicated to interphase. This is the prep work, the behind-the-scenes stuff. During interphase, which biologists break down into the G1, S, and G2 phases, the cell is intensely active. It's growing, it's pumping out proteins, and critically, during the S phase, it's synthesizing and completely copying that entire 2 meters of DNA we just talked about. A mere 10% of the cycle is the mitotic, or M phase, where the actual physical division finally occurs.
But it is also absolutely crucial to understand that not all cells stay on this continuous track. Lots of the highly specialized cells in your body right now, like your fully formed mature nerve cells or your muscle cells, they actually exit the cell cycle entirely. They shift into this permanent non-dividing state called the G0 phase. Basically, they've retired from dividing. They are very much alive, functioning perfectly well, doing their specific highly complex jobs, but they are totally out of the replication business.
All right, now for the cells that do divide. Let's look at Module 3, the dance of mitosis, focusing on the incredible choreography of cellular division. Mitosis itself progresses through 5 distinct sequential steps. You've got prophase, prometaphase, metaphase, anaphase, and telophase. Basically, the cell goes from the initial condensation of chromatin to the fragmenting of the nuclear envelope, to lining up all those chromosomes right down the middle, then the sudden, rapid separation of identical sister chromatids, and finally, the arrival of complete chromosome sets at the opposite poles of the cell. It's a beautifully synchronized, flawless routine.
But here's the really crucial question. How do those chromosomes actually move around during this dance? The unsung hero of this entire process is a structure called the mitotic spindle. This is an intricate web of microtubules that forms right inside the cell. During division, these fibers reach out and attach to specialized protein structures on the chromosomes called kinetochores. Then, motor proteins literally walk the chromosomes along these microtubule tracks. Scientists actually often describe it as an arcade Pac-Man mechanism. The motor protein literally chomps down and depolymerizes, or breaks apart, the microtubule track behind it, reeling the genetic material safely to the opposite poles. It's totally mind-blowing when you think about it.
To finally finish the division process, the actual cytoplasm itself must be split, which is a process known as cytokinesis. But this looks entirely different depending on what kind of life form we're looking at. Animal cells use a contractile ring of microfilaments that acts exactly like a drawstring, pinching the cell right in half to form what's called a cleavage furrow. Plant cells, on the other hand, have rigid cell walls, so they can't exactly pinch. Instead, they send these little vesicles to the middle of the cell, which coalesce and amazingly build a brand new structure, a cell plate, right down the center, dividing the space neatly in two. It is just a spectacular display of cellular construction.
Let's transition into our fourth module, the checkpoint control system, where we examine the strict security regulating the cycle.
We have to ask a really profound biological question here. What dictates the pacing of all this? What exactly ensures that a cell doesn't just start dividing completely out of control? Well, biologists often compare the cell cycle control system to the internal dial of a washing machine. The cycle proceeds based on an internal clock, fueled by the rhythmic rising and falling concentrations of specific proteins. The main players here are proteins called cyclins and enzymes known as cyclin-dependent kinases, or CDKs. When these guys combine, they form complexes like MPF, which stands for M-phase promoting factor. Think of MPF as the ultimate molecular go-ahead signal. When MPF levels peak, it triggers the cell to physically launch into mitosis. Once the job is done, the complex just breaks down naturally, effectively resetting the dial for the next cycle.
But this internal washing machine dial isn't flying blind. It has highly sensitive internal sensors that report back on whether crucial processes have actually finished. For instance, right before the cell splits apart in the M phase, there is a very strict checkpoint. The cell will completely halt the division process if even a single chromosome isn't perfectly attached to the spindle fibers. It literally just waits, delaying anaphase until every single kinetochore is locked in securely. This is a brilliant security feature because it guarantees that the new daughter cells don't accidentally end up with missing or extra chromosomes.
And normal, healthy cells are also strictly governed by external physical rules. First off, they exhibit density-dependent inhibition. Simply put, if cells get too crowded and start bumping into their neighbors on all sides, they send signals to stop dividing. I mean, nobody likes a crowded room, right? Second, they have anchorage dependence. A normal cell absolutely refuses to divide unless it is physically anchored down to a surface or a tissue matrix. These strict rules keep cellular growth highly organized and contained.
Which unfortunately brings us to our final module today, Module 5, When Cells Go Rogue. We need to talk about the chaos of cancer.
We're going to look at the terrifying chaos that unfolds when the very controls we just studied break down completely. Cancer cells simply do not heed the normal signals that regulate the cell cycle. Because of genetic mutations, they essentially become blind and deaf to the rules. Unchecked by that density-dependent inhibition we just talked about, they don't stop growing when they get crowded. Instead, they just pile up into a massive abnormal cells called a tumor. And because they ignore anchorage dependence, they can lose their attachment to neighboring cells, slip into the bloodstream or lymph vessels, and invade entirely new tissues. This is a highly dangerous process called metastasis. They are, at their core, cells that are dividing with absolutely no off switch.
Because cancer is fundamentally a disease of the cell cycle, our traditional treatments are actually designed to weaponize that very cycle against the tumor itself. High-energy radiation damages the DNA of cancer cells, which, unlike healthy cells, often lack the ability to repair it. Then we have chemotherapy drugs, which are introduced to the bloodstream specifically to attack actively dividing cells. Take the drug Taxol, for example. It works by physically freezing that mitotic spindle we talked about earlier. It stops that Pac-Man mechanism dead in its tracks, which destroys the dividing cancer cell. But here's the catch. Because it targets any actively dividing cell, it also deeply impacts normal, healthy cells that naturally divide frequently. And that is exactly why patients undergo such difficult side effects like hair loss and nausea. We're hitting the good machinery along with the bad.
But let's look ahead, because our growing understanding of this exact cellular biology is leading us away from those blunt tools of traditional chemotherapy and guiding us toward the incredible frontier of personalized medicine. Today, by quickly sequencing the DNA of a specific patient's tumor, we can actually find the exact broken checkpoint or faulty receptor.
For instance, if a breast cancer tumor has abnormally high amounts of a receptor called HER2, we can now prescribe targeted molecules like Herceptin to block that specific rogue signaling pathway. We aren't just carpet bombing the body anymore, we are fighting the broken cellular machinery directly at the molecular level.
So, as we wrap up today's masterclass, I want to leave you with a really thought-provoking question based on everything we've covered. As our technology continues to improve, and as we master the absolute ability to molecularly reprogram a cancer cell's broken checkpoints and silence those rogue proteins, how dramatically could we alter the future of human medicine? Could we fundamentally change the human life cycle itself? Think on that. Thank you so much for joining me for this explainer, and as always, keep learning.